Hello, everyone, and welcome to today's webinar titled Strategies for Epidemics Containment in the Context of the Anthropocenes Planet Degradation. This webinar is part of the Microbiology Virtual Week, and I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots. So let's get started. Before we begin, I want to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. Now, you can find that live chat located on the right of your screen. You can also participate by submitting as many questions as you want during this presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. Now, if you have any trouble seeing or hearing this presentation, simply click on that help desk button located at the bottom of your screen within the navigation bar or from the lobby. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on that continuing education credits link located in the abstract window below the presentation window and follow the process to obtain those credits. I now present today's speaker, Dr. Francisco Veas, Research Director and Professor at the French Research Institute of Development at Montpellier University. For a complete biography of our speaker, please visit the Biography tab at the left of your screen. Dr. Veas, welcome. You may now begin your presentation, sir.
Okay, first of all, let me thank you this opportunity to share some ideas and some results that we have generated during this COVID epidemic. Let's go for the title here. Then, strategies for epidemic containment in the context of Anthropocene planet degradation. As everyone knows, we are following temperature elevation of the planet, and then and many others effects generated by humans. This is because I'm talking about Anthropocene, which is a period comprised from 200 years ago, at the beginning of industrialization, and now. Because research or whatever be the activity that we can do, medical activities, agriculture activities, and so on, whatever, everything is being done in a particular context. In people that works in tropical environments or other environments, whatever, in the US, in Europe, and then all of us in the planet are submitted to this context. Then this is because I wanted to talk about this first slide with this particular title. Then let's go to a part of introduction just to continue to talk about that. Then the planetary context is as follows. We are following a high pressure and rapid loss of life, living beings, let's say, even geophysical changes, for example, ecosystems, biodiversity, human demography. All of these aspects are changing our planet. For example, here on the top right of the slide, we have a simple comparison between human population in 1800 and now, 2050. Then we have a difference of 1 billion in 18,000 and 10 billions in few years from now. Then that means that we will be 10 times more in a very short period of time, 200 years, let's say 250. And second, we have a lack of the capacity of international healthcare community to provide efficient, timely response to infectious outbreak. This is the situation. You can see in the figure on the top, on the bottom right of the slide, numerous outbreaks, including Ebola, Lassa, Zika. Now, for preparedness and surveillance for improved responses to re-emerging pathogens or new emerging pathogens, we need to think to act in a totally integrative way, to consider always the whole planetary infectious context. This is the situation that we are living now. And then our idea is to provide efficient solutions through conceptual and technological preparedness, as well as obviously an accurate surveillance of these pathogens. Then this needs heavy investments, in particular on at-risk approaches, in order to rapidly explore innovative solutions, as scientists did for RNA vaccinations against SARS-CoV-2. This was very rapid response, very good. Even if not a sort of panacea, this solution is permitted to the human population to control this epidemic as much as we could. Then, for carry out the surveillance, we need obviously clinicians, veterinarians, and environmental actors in the field, as well as efficient ultrasensitive for early pathogen detection, in order to adopt adaptive countermeasures. In particular, for example, for this kind of diseases like Ebola, monkeypox now, and to detect also residual viral loads in convalescent individuals that could reinitiate an epidemic. It is important to detect very early to stop the spread of the viruses or pathogens in general, and obviously residual viral loads in a way to be sure that people are free of pathogens to be released in the nature, let's say. Okay. Then from this situation, I think that we can discuss about the fact that we need to learn some lessons from this zoonotic SARS-CoV-2 COVID pandemic. In multiple cases of infectious disease outbreaks, human has indirect or direct responsibility. For example, through the intensive animal farming for human consumption, massive of rare or protected animals for human consumptions, illegal, in particular in wet markets in Asia or in Africa. This is very frequent. Or another different reason is poorly justify hazardous creation of pathogens with gain of function, totally ignoring, in this case, consequence and obviously the mitigations needed to stop what these researchers could create. I will explain a little bit with some examples here. For example, the pig farming has generated in 2009 flu H1N1 outbreak that took only 6 months to spread from Mexico to the whole planet. By chance, this was a low, moderate transmission, low pathogenesis, and then the epidemic stopped 6 months later. Another case of poultry farming, in this case, was the avian flu, highly pathogenic one that was killing some humans, not that much by chance, 800 all over the world. But the point here that this virus was transformed to gain function for a higher transmission among mammals. That means among humans, and this was done by European researcher in Netherlands. And then, because they published this data in Science, then they generate the idea to other researchers in China to generate more than 100 GOV H5N1 and H7N9. And then that means that we have in the fridge, in somewhere, very dangerous viruses that could escape from those places by accident or whatever be the reason. And the last example is SARS coronavirus variants of concern, that mean alpha, beta, Omicron, BA5, and so on, different variants that were originated from Wuhan in China, respiratory virus with high transmission capacity. The problem that we had here is that Chinese healthcare authorities didn't timely transmit critical information to WHO in order to adopt mitigation countermeasures. And this is the kind of thing that we need to avoid in the future. Despite the WHO-driven international, let's say, incomplete investigation in last year, we lack lab books, electronic data were erased. More than 2 years, that means now we don't know, we still ignore the virus origin. And this is a problem because when we have more information, we have more tools we can develop to better respond to this kind of outbreaks. As consequence, this COVID-19 has generated over 6.5 billion human deaths and over 600 millions of direct victims. By chance, most of them recovered with because this is let's say a nice virus, is not so pathogenic, despite all the cases that we know. And COVID has massively impacted also socioeconomic aspects and generated dozens of trillions of dollars of loss for the worldwide economies. Then the impact of this SARS-CoV-2 has been quite strong in economical terms. Thanks to the first historical severe prolonged worldwide lockdown of human populations, this has generated a dramatical decrease of human activities during this period. Then, from that, we have two kinds of effects, positive effect and negative effect. Indirect, highly positive effect of COVID on the health of the planet. We see unprecedented decrease of manufacturing, mining, agriculture, fishing, transportation, pollution, including plastic, fossil resources, toxic gases, noise, and light. We had also the possibility to see that a daily decrease of fossil dioxide carbon emissions that were reduced by minus 17%. That was compared at the levels of 2006. Very, very important then. And in 2020, the growth rate of the global population fell under 1% per year. This is have never seen since 1950. Then these are, let's say, positive effects. And we can see on the right side a photograph, a satellite photograph to detect nitrogen, dioxide nitrogen, sorry, in China, in the region of on the east of China, and then we can see that before in January, before the lockdown, you have an intensive presence of this nitrogen dioxide. In contrast, soon after the start of the lockdown, you have immediately almost nothing. That means that the toxic gases can disappear very quickly. And then this is another very positive effect. As negative effect, we can see that poaching was one of the negative effects in Africa and Asia that reduced biodiversity. They were chasing pangolins, rhinos, elephants for Chinese markets, and other wild animals for food. Then markets and food because they were lacking food because of the lockdown. Then these are negative effects. Then let me show you some approaches to better prepare surveillance of re-emerging diseases in order to better adapted countermeasures. Then let me show to do that, working on SARS, we have demonstrated that ultrasensitive detection of pathogens is mandatory for an efficient surveillance to early contain pathogen transmission, reduce false negative diagnosis, and detect residual viral loads in convalescent to prevent any new epidemic ignition. One of the key strategies to reach this objective is to realize a sample preparation allowing pathogen concentration prior the step of pathogen detection using classical PCR, RT-PCR, LAMP, etc. To give more chances to detect low pathogen loads in complex samples. Then the SARS-CoV-2 example will be seen here. But before, let me show how we did to reach this objective. Then we use a protein which is called apolipoprotein H, which is a pleiotropic protein from the plasma belonging to the innate acute phase response. This is, let's say, one of those proteins, like CRP, which is the most well-known, from the first line of immune response in our bodies. And the role of this protein in this context is a sort of sensor and scavenger of non-self. Then it can catch any kind of pathogen, but not commensal bacteria, for example, from the gut, because it detects only pernicious microorganisms. This is a small protein, around 50 kilodalton, in a concentration close to 200 milligrams per liter in plasma, able to interact with high affinity at nanomolar scale, with electrostatic interactions, hydrophobic interaction, protein-protein, and then it could react with LPS from E. coli, phosphatidylserine from HIV, HCV, and other viruses, SBI or M1 from other bacteria. And then on the right side, you have the, let's say, a representation of the structure of this protein with 5 regions showing different regions that could react with different molecules like LPS or another phosphatidylserine, etc. And then we are able now to produce these derived peptides that mimic the ApoH functions, and then these mimetic peptides from ApoH are able to interact with all these pathogens as is as needed. Then what we do is to put these peptides on to coat, let's say, surfaces or magnetic beads or membranes or chips that will be used to concentrate the pathogens before to be detected. I will show you how. Let me go here. Then you can see on the right, on the left side of your screen, the beads on the upper side of the tube coated with ApoH peptides, and the tube containing pathogens, viruses or bacteria, whatever. And then what we do is put these beads into this sample, complex samples, can be serum, saliva, any biological or environmental sample. Then we agitate for a few minutes, and then we apply a magnet that will catch the magnetic beads that has already captured, and we are going to concentrate on this following step by precipitating all the beads that has the viruses captured. Once we are in this step, we are ready to detect what we want to detect. We can sequence, we can do PCR, or we can isolate by cultivating these captured pathogens. We can use any kind of detection system, for example, bioluminescence or bacteria, etc., etc. Then this is a very simple way to concentrate prior the detection, that giving us the opportunity to detect very low viral loads or bacterial loads from a complex sample. And then if I move correctly, and then this is an example where we did an isolation of hantavirus from Andes, highly pathogenic respiratory virus. As you can see, we isolated the strain 1 and strain 2, the new isolated strains, as compared with, on the left, you have the phase contrast on the microscope, an antibody that recognizes only the N protein of the virus, and then on the right side you have the nucleus of the cells. Then you can see that the lab strain was perfectly captured, and in the absence of the virus we cannot see the green color. In contrast, we can obviously see the new isolated strains 1 and 2 using this procedure. I can show you also another aspect that we need to know is ApoH bind and capture only infectious non-self, in this case viruses. This is the case of dengue. Then when we did an experiment where we took the sample and we took the sample normally, and the other sample was heated 1 hour at 56 degrees. And then we were able to denaturate the surface of the dengue virus. Then when we proceed to the dilution of this virus here in the graph, and the different situations in the presence, in the absence of ApoH is in blue, or in the presence of ApoH, we can see that we capture everything when we don't dilute the sample, or we dilute only, let's say, 10 times, 100 times, 1,000 times, etc. And we can see that we are able to detect at highest dilutions only viruses with ApoH and not without, in blue, here in gray. And obviously, viruses that were denaturated, we were not able to be detected at the highest dilutions. Then this is an important aspect to know, because what we need to carry out these kind of technologies is to have high quality samples. For example, samples that are frizzy and thawed many times, many cycles, are not adapted for this kind of studies. Oh, sorry. Here I can show you what we observed in the case of hepatitis C at the hospital, from negative 25 negative samples that were suspected to have hepatitis C. And then in the hospital, they realized one of the reference detection system using COVAS RT-PCR for HCV. They detected seropositive in those suspected samples. Instead, they found obviously 25 negatives, because they were only suspected and the COVAS was negative. Instead of that, ApoH plus this RT-PCR, we were able to detect 11 out of these 25. That means that we are avoiding these false negatives. And this is very important to adopt the proper countermeasures or treatments or whatever we want to do with those patients. Then has direct consequence for the patients, the false negatives. Here we did something comparable, but this time with Ebola. And then you can see here the samples. Here you can see that normal detection done with the qPCR done by use and breed with their own FDA approved diagnostic. There were one sample negative that were positive when we use ApoH systems. Moreover, if you look this column as compared with other columns, we have a gain of 3 CT of amplification. That means that we have at least 1 log more sensitive detection of Ebola. And this is very important, in particular when we are in presence of convalescent patients, as we did here, for example. Convalescent patient that we were having reads because we did sequence and PCR. We had 3 logs difference. And moreover, doing sequencing, we were not only to detect Ebola, but also some co-infections with Lassa virus, which is a confounding hemorrhagic factor. Then you know I want to show you that the way to concentrate low viral loads that are not detected with the current existing reference systems is a very important manner to better control epidemics and better manage the patient treatments. And when we did some environmental flu detection, we can see that in the presence of ApoH here in pink, we have a difference 1,000 times the sensitivity to detect reads with ApoH as compared with the no with the absence of ApoH. Here, much more, almost 1,000 times, because we go from sometimes from 0 to 1 million or 1,000 to 1 million. Then the enrichment is considerable. Here, very quickly, the same for influenza virus. I don't want to bother you too much. Just show you that whatever be the dilution here, I don't know what happened with the. Okay, sorry. Okay, here dilution 10 to 10. Then you can see that with ApoH we can really be much more sensitive to detect any kind of virus. And one example, very short, with the sepsis bacteria. This is Staphylococcus aureus, that where we could show the detection only in half a day, and able to detect in 5 ml of whole blood only once one colony forming unit of bacteria by PCR. These results were confirmed with larger cohorts by Siemens that published this data several years ago. And then you can see that this system works for many type of viruses or bacteria. In the case of sepsis, this is very important because it's one of medical emergencies that need to be treated very, very rapidly. Otherwise, the patient can die. And then we need to provide to these patients an appropriate antibiotic strategy. Then the next is just to show you that many bacteria, we can work with many bacteria, including some bacteria here as Trophirma whiteplayi in the right side in the bottom, which is a bacteria that is very difficult to cultivate. It takes 15 days. It can be detected within a few hours when we treat with this ApoH system. Let me then show you what we did with very rapidly with COVID using this system. We taking the property of concentration of this ApoH and the viruses. We use it as invasive and self-collectible sampling for an ultra-sensitive detection using RT-PCR. Then if we move, we can see that this is the process. We can collect different oral, gargle, nasal sampling at home, some of them, or in the hospital for others. Then we use the system magnetic beads with ApoH, and we the gargles were treated, for example, with 10 ml of water that were put in presence of with the beads for 5 minutes agitation, and then the separation with magnetic system, and then the concentration just before the PCR that is being done here. And then using this, using this protocol, we can see here in the next, for 85 persons in the clinical trial, we have shown that red compared with blue is without ApoH, here is with ApoH. Then whatever be the gene that you are using to detect the virus, always is in favor of ApoH, with a very important gain of sensitivity, almost always 10 times. In addition, you can see the gray zone where the virus are not detected with the blue system, the normal PCR, let's say. They are always detected with ApoH. That means that we can face the false negative generated by the normal detection manner. Then if I move to the next, and then here the most important is to show that this approach works for all the variants of concern. Here we work with alpha, beta, and gamma. Then you can see with ApoH is much better with that without in gray. And the next is very interesting because I will show you a follow-up in 3 patients where one was positive at the hospital, and then we did the follow-up with the system ApoH. It was always positive and always gaining 2 to 3 CT, sometimes more. And then this was also a positive that was followed up to the end of the infection. And this was negative at the hospital, different days. Then this is a false negative because it was positive with ApoH. Then that's it for this system, that ApoH and viral concentration, when we use non-invasive, can be very useful for ultrasensitive detection, say 10 times better than the RT-PCR alone. We reduce the false negative, as I told you. This is simple, rapid, and well adapted for diagnostic in children that don't like the nasopharyngeal approach, or personnel that need repeated virus control in the hospital, for example. This can be easily used also with isothermal amplification, not only PCR. And then what we are doing right now is sequencing, and can be used, as I showed you before, like a motor of discoveries. Now we are presently working in Aurus detection system concept, which is means advanced ultra-rapid, ultrasensitive detection kit to adapt it to different panels of pathogens, SARS-CoV, monkeypox, bacteria, whatever. That means a test that can be done very sensitive way, very rapid, in less than half an hour. Then this is a sort of solution for what is coming. Because of the situation of the planet, we are going to lack water, and the situation will be highly degraded. Then we need to face the next outbreaks with simple, rapid, and efficient solutions. Now we have some, let's say, some times to act, and we have a lot of means, but because the situation will be degraded, then we need to think about new solutions. Let's move to the end of this talk. Let me talk to you very quickly about the global context that we are following. Then what we see, the big story of complexity from the Big Bang up to the apparition of life 4 billion years ago. Now we are in the Anthropocene that started 2 centuries ago. Then this Anthropocene is clearly severely impacting the planetary geophysical and living conditions. Geophysical because the water, because the temperatures, because the polar calottes that are being thawing. Multiple critical planetary parameters undergo radical changes. In particular, in these last 40 years, we can see that we have simply lost 70% of vertebrates, 8% of insects, and 50% of vertebrates, birds. And all the rest, 30%, 20%, and 50% are in extinction way. In particular, due to poaching and industrial fishing, etc. Multiple human activities negatively impact the ecosystem, include the extraction and use of natural and fossil resources, mega fires that we know in several places of the world, in Amazon, where man create these mega fires. With pollution, we reduce the natural ecosystem, habitats, wetlands, mangroves, glaciers, polar caps, rivers, lakes, and we know everything here. In Colorado, the Colorado River, you know that is suffering a severe drought now. Then, moreover, we are increasing the terrestrial and ocean temperatures, mainly due to dioxide, carbon, and methane. All these data are correlated with uncontrolled demographic growth. Since 20th century, we are reaching the 8 billion humans now this year, as compared with only a single billion in the beginning of 19th century. But the point is that if we compare 2022 with 1800, one human from now use a lot of resources as compared with a human in 1800. Then, because this corresponds to 1,000 times more environmental impact, we are now, as compared with the people 2 centuries ago, we are not 8 billions, but we are 8 trillions of humans in terms of consumers. And then you can see the impact that we could have, or we are having on the planet. This is one of the contexts. And this was the increase of the population was due to the antibiotics that were used after the Second World War, even if they were discovered very early before, because vaccines, therapeutic strategies, and this has contributed to unprecedented demographic growth. Then this is a paradox of the medical progress that could accelerate, by the way, the planet degradation. Then we need to find solutions. We need to be more intelligent than we are now, because this situation is making that pathogens are concentrating in highly dense, promiscuous habitats, increasing the severity and frequency of epidemics. Then you see the context impact the consequence. And then, if you know Intergovernmental Panel for Climate Change, IPCC report 22, they show us that that impacts on climate change are observed in many ecosystem and human systems. As you can see here, high or very high impact on changes of ecosystem structures, whatever be the place, Africa, Antarctic, etc. You can see that everything is negative. In freshwater production or oceans, is always negative. The species range shifts, terrestrial, freshwater, oceans. On the species also, the impact is very negative, in dark blue here. Changes in timing, then terrestrial, this is the cycle of those ecosystems. Then everything is being impacted highly negative because of the human action. Here, observe impacts of climate change on human system, impact on water scarcity and food production. Then everything is negative, or for the moment, we cannot see if there are sometimes a little bit positive aspect, but most of them are negative, as you can see. Fishing production, poultry, etc. And then impact on the infectious diseases, there will be more and more, and so on. I don't know. You can see this in the IPCC report that is available on the online. And then they propose this graph that show us all the negative aspects that we can see, and some possibilities for resilient restoration of living conditions. If we do act quickly with different aspects that I will show you here in the next, you can see that we can maybe keep the temperature below 2 degrees. 2 degrees for the mean of the planets will induce crazy things, totally crazy things, as this report is reporting. Then some examples to make positive, but we need to be active right now, immediately, no room to waste time. First of all, we need to learn and train designers and broadcasting media, because they ignore most of the things that we are talking about. To calculate the real price of things, considering the huge nature added value. For example, water. There are companies that use billions liters of water to produce, for example, Coca-Cola, Pepsi, or whatever producers of water. They use and pollute a lot of water that they don't pay the price. We need to create new jobs related with restoration and resilience of ecosystems for young people, old people, whatever. Everyone not need to be involved. We need to reduce the world populations with policies reducing fertility, reduce transportation with fossil energies around the world, reduce to 3 tons of CO2 per individual, because in France, for example, we consume, in the mean is 11.5 tons per individual in France. Then we need to reduce almost by 80%. We need to replace all the fossil energies. We know something here in Europe because of the war in Europe, Russia and Croatia. The Russian war, and the one important and easy thing that we need to do is to plant 1.2 trillion trees, in addition to existing 3 trillions, not billions, 3 trillions, to restore the other 3 trillions that we destroyed already. To both, these 1.2 trillion trees will absorb almost all the exceeding of carbon dioxide for the next 10 years and save essential strategic tree species to maintain life on the planet. These are simple, some of them are simple things to be done. Others are not so simple, but we can do that. I don't know. I don't know if you can see this screen. I have a big screen on my computer. Then conclusion. The pandemic experience must learn us something, and then we must listen to this experience. We have a lot of consequence of this Anthropocene, the 6th mass extinction that we discussed already. Despite multiple alerts that has been provided by great thinkers as Prussian French naturalist von Humboldt, Alexander, in 18th century, the economist Thomas Malthus in 18th century, Paul and Anne Ehrlich from the last century, and right now they are continue to alert about that. Philosophers from the 16th and 17th centuries, Montaigne, Spinoza, Friedrich Nietzsche, in the 19th century, including volcanologists, astrophysicians that started with Copernicus, Galileo Galilei from Florence, Carl Sagan, Aurélien Barraud in France, now in present, all of them tell us, hey guys, move ahead against this big catastrophic context that we are living. By contrast, we were unlucky to have this Descartes in France in the 7th century that promoted the wrong idea that the nature is a machine to serve humankind. He considered humans to be at the highest evolutionary position out of the nature. And the error is that we are in the nature. We belong to the whole ecosystem. Consequently, considering the holistic planetary context, we need to accept the maximum of intelligible information that we can compute from different situations, healthcare, etc., to rapidly generate rational and intelligent solutions. Despite all the negative aspect of COVID is learning something to us. Most of the infected patients, there are some positive also aspects, are asymptomatic. The high majority of symptomatic patients recover. Induce low moderate mortality, only 1%. This acute infection is easy to potently be neutralized as compared with HIV or dengue or other viruses, for example. And then using simple or new vaccine strategies, virus can be neutralized. Or as well as with host polyclonal antibodies, the neutralization is very high. As you can see in the figure on the right side, the serum needs to be diluted between 10 to 5 or up to 1 million times for different kinds of variants, including one of the latest one, BA4 and 5. And this is being done in a framework of European project called EPIC-Ground 2 consortium. And then I will finish with this. This virus had the incredible power to stop for a few weeks the crazy race of humanity to go anywhere, demonstrated that rapid and substantial environmental restoration is yet possible in the absence of humans, that most of human activity that most of the time are not really needed. Therefore, instead to neglect and rapidly come back to our previous way of life, as humans are doing after the lockdown, this planetary real-time demonstration, COVID demonstration, must be highly be considered as a guide to rapidly start to restore our wonderful oikos, planet, our wonderful home, let's say in Greek, for its destruction resulting from the reward dependence of our brains via the striatum dopamine axis. We can talk about that if you want for some questions. I want to point out that discover something via a rational, intuitive, or counterintuitive thinking, like Galileo or Einstein did, or simply by facing serendipity situations. This latter is only possible if the discovery is able to see the eureka moment due to the analogic capacity of the brain to make necessary connections and resulting deductions, as did young Fleming in 1928 to discover penicillin from a total unexpected situations. Microwaves were discovered like in that way, serendipity discovery, as many other discoveries that did in that way. And then my final comment will be, consequently, we cannot ignore the COVID-19 lessons, and we need to see this eureka moment. Since we need to start to set up new existential paradigm compatible with the beauty of life that we see the destruction right now. Thus, we must plant trees, as I told you, 1 billion point two to reduce CO2 and produce O2, consume much less, in general, animals in particular, give more space to wildlife, will dilute pathogens instead of concentrating them otherwise. We need to reduce demography and blah, blah, etc. I would like to finish here, despite my huge screen. Thank you very much. Then I take a question if we have time. Thank you. I think that I was over the time, but sorry for that. Thank you.
Absolutely no problem, Dr. Veas. Thank you very much for that informative presentation. And we will now start the live Q&A portion of the webinar. If you have any questions, please ask now. Just click on that Ask a Question box located on the far left of your screen. And we do have just a little bit of time. I'm going to ask 1 or 2 questions and see if we can get them in, Dr. Veas. Our first question: can you please comment on consequences of false negative diagnosis in COVID-19 and other diseases?
Yes, this is very common because all the diagnostic system existing already, or which are reference diagnostic systems, FDA approved, etc. I am talking of high quality system of detection. Most of them, when the pathogen loads is too low, let's say, they are not able to detect the presence of pathogens in the samples. Most of the time, samples are too small, and then these existing tests work with very small quantities, at the most 1 ml at the maximum. And then obviously we need to develop systems that are able to detect even 1 pathogens per ml or per 5 ml, as we did for sepsis in whole blood. And that means that the impact is double. One for the patient, if you are considered as a negative person, that means that if you were infected, the doctor will tell you, oh, okay, you are cured already, then feel free to to go, and then you will not be provided with a adopted treatment and whatever. Then this could be pernicious for your health. This is for the individual, and for when we need to contain some epidemics like Ebola, for example. If some patients go out of the center of treatments of Ebola, and then these people that are in fact not negative but positive will spread the virus on the population, and the epidemic will grow, as we knew in the last big epidemic in 2014, where we had a lot of contamination due to the false negatives.
Dr. Veas, thank you very much for your time today. We're not going to be able to answer any other questions. What a great presentation you have, and thank you for your important research. Before we go, I do want to thank our audience for joining us today and for their questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand for 2 years until September 8th, 2024. LabRoots will alert you via email when it's available for replay. And we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Bye-bye.